But simply stated, in the presence of the PPAR gamma activators or ligands like indomethacin, ibuprofen, and DHA, in the experimental studies using rat cortical neurons that are cultured, typical control, when you add in the amyloid, the number of neurons that survive goes down dramatically. But in the presence of these PPAR gamma activators like indocin and ibuprofen, there is survival of the neurons. Collectively, the results indicate that these types of drugs are very effective neuroprotective agents, anti-inflammatory agents, and ultimately may play a role in such disorders as MS, Parkinson's, Alzheimer's, and other inflammatory conditions, and we'll look at them. Briefly, when you give MPTP to an experimental animal, you damage the substantia nigra. You produce Parkinson's disease or a Parkinsonian type of response in that experimental animal. And this has been the prototype now for studying Parkinson's ever since MPTP was discovered as a synthetic Demerol analog developed in California for people to get high, and suddenly the next morning about 15% of those individuals exposed to this neurotoxin came into the emergency rooms with flagrant Parkinson's disease. So while it was an awful thing to observe, it provided an incredible bit of information about Parkinson's in terms of an experimental model. Basically stated, when MPTP is added, the number of neurons surviving in the substantia nigra pars compacta was dramatically reduced in the presence of this neurotoxin, but in the presence of pioglitazone, which is the anti-diabetic drug Actos, used by millions of people, those neurons survived. Pioglitazone may offer a treatment opportunity in Parkinson's to slow the progression of the disease that is mediated by inflammation. So what's happening now is we are seeing people begin to focus on treating the disease, not just the smoke, but treating the fire. You know, sometimes when I lecture to groups of Parkinson's patients, I'll say, how many people in the audience are taking a medicine to treat your Parkinson's disease? And the hands go up, albeit somewhat slowly. And I said, well, if you're taking Sinemet or Requip or Mirapex, I'd like you to put your hands down, because you're really not treating your Parkinson's, you're only treating symptoms. And treating the disease is really the focus. And they all look surprised. Actually, Parkinson's patients don't look surprised, but they're a tough group to lecture to. Another group is, you lecture to a group of obsessive compulsives, and then you get letters afterwards, and they come on Tuesday every week thereafter, you know. In the animal model of ALS, we see that, again, it's an inflammatory issue. And in this case, this transgenic superoxide dismutase mutation, a model of ALS, again, looking at the effect of pioglitazone, a dramatic increase in the number of muscle cells and neurons that survive, to the extent that these animals actually, to some extent, actually improved. And their conclusion is, because the drugs concerned have been proven to be safe and already in clinical practice, they would be ideal drugs to consider using in these neurodegenerative conditions. Well, you might ask yourself then, we're not here to talk about pioglitazone or pharmaceuticals, but what are nature's PPAR gamma ligands? I'll give you a hint. It's a 6-weight fly rod, sinking line, and this is Alaska. All right. Very few people got that, but we'll talk after. It turns out that when you look at the metabolites of DHA, and this is how these are the R groups and how they are substituted. The second metabolite of DHA shows an incredible activity in terms of serving as a PPAR gamma ligand. DHA, the second metabolite, is actually almost twice as powerful as a PPAR gamma ligand as Actos is. So, I mean, it's not as if this biochemistry evolved to be where it is so that we could develop pioglitazone. I mean, our task is to try to go back and unravel this information. You know, in Naples, Florida, I'm often accused of practicing non-traditional medicine. This is how medicine's understanding, you know, of reducing inflammation by the foods we eat is certainly the more traditional. In looking at autism, which is a brain inflammatory condition, very briefly, we see that children, when we reduce inflammation by giving them this drug Actos, their hyperactivity decreases, stereotypic behavior, lethargy, and irritability. This is an interventional trial giving pioglitazone or Actos to autistic children. Now, you individuals who are treating autistic children using high-dosage DHA now should really understand that the biochemistry behind what you're doing has a lot to do with this PPAR gamma ligand activation, therefore reducing inflammation. Well, what regulates, what governs this whole PPAR gamma activity? And this calls to us this whole sirtuin information. Sirtuins are proteins that protect DNA. And in so doing, in protecting DNA, sirtuins are looked upon as being possibly the Methuselah factor, enhancing lifespan. This is hot science right now. They protect DNA because they acetylate the histones and allow the DNA to be more compact and more protected against being damaged by environmental issues. Studied dramatically in fungi, and also in worms. So this is really front and center right now. People are talking about sirtuin chemistry and what it is that activates sirtuins in the worms and other organisms. One of the most powerful initiators or activators of sirtuin chemistry is caloric restriction. So this is what seems to be activating this whole pathway in animals. Why would caloric restriction lead to longevity? Work by Dr. David Sinclair, published in the Scientific American, Dr. Sinclair from Harvard. Why is it important to extend our longevity? What's the correlation between caloric restriction? Well, quite simply, if you can't find food, you're going to need to live longer to find food in order to reproduce. Well, this whole idea of restricting calories or fasting these animals or humans to extend lifespan, this is incredibly new information. It was only just described 4,000 years ago. The Sanskrit word upavasa is the Sanskrit term for fasting. And what does it mean? It means returning to life. So, our task seems to be to try to understand what's been known for a long time. I mean, how did the Chinese know that ginkgo biloba was good for the brain? Where does that knowledge come from? And I've been participating in a little bit of caloric restriction lately in high-dosage DHA. And I'll tell you, it makes you very, very sharp. It makes you smell things in a more refined way, taste things, experience things. And a couple days ago in Sedona, I was walking through, we were doing some hiking, and this whole concept of, when the tribe is walking through the forest, how do they know what to eat or what not to eat? And I had a thought that perhaps that knowledge is there. Perhaps that knowledge is in our DNA, that we would know which plants we should eat. I mean, the concept is there's always one person in the nomadic hunter-gatherers that gets elected to eat whatever it may be, the hemlock today, and that he or she then eats it and he's done, or she's done. But I submit that information's in our DNA and that over the tens to hundreds of thousands of years of our evolution, it's there. And we just have to become more closely adept at identifying it and understanding it and putting it into practice. Well, so basically the concept is that caloric restriction is life extending, but more specifically for our purposes, caloric restriction attenuates the production of beta amyloid. This sirtuin chemistry has a profound effect on the brain and on amyloid production. So, this whole idea of fasting was talked about this morning. What was it, 180 days that they reduced their caloric intake on Crete? Well, it spans all cultures and all religions. Everywhere from Ramadan to Yom Kippur, there's a fasting holiday or more. So the other interesting thing that we are now learning is that another powerful activator of sirtuin chemistry is resveratrol. And this is, again, why resveratrol is so front and center right now. And studies that are showing resveratrol reduces inflammation, reduces amyloid production, are really becoming very important because they call our attention to the fundamentals of inflammation and ways of getting to it. Some of these slides you will not see. They were just added because I recently took a trip to Peru, my family and I. And in Maras, we met this gentleman who gave me some understanding about genes and genetic expression that I think was there but really needed some explanation. We started off in Maris in this Inca laboratory. We were touring with Alberto Villoldo of the Four Winds Society, who, oddly enough, he and I are writing our next books together. He is a medical anthropologist who visits with the shamans, and I'm a neurologist. So where we come together is going to be an interesting place.